With that, I'd like to introduce Dr. Alex Spira from the Virginia Cancer Specialists and VCS Research Institute, who will tell us how immunotherapy works. Alex.
Thanks. Good afternoon. So immunotherapy, relatively new over the few years, and its role in cancer goes back several years. In medicine, we've been noticing that for a long time that people having problems with the immune system often develop certain kinds of cancer. Certain groups, such as organ transplant groups, people taking immunosuppressive drugs, people born with AIDS, people with HIV, and other rare medical syndromes were very prone to malignancies. And with that in mind, we understood for a long time that the immune system kept cancer in check. With that in mind, a few years ago, the roles of cancer immunotherapy came about to really treat malignancies and come up with a new mechanism of action after chemotherapy. On the slide, you can see there are several different mechanisms of action. The earliest are these monoclonal antibodies. Monoclonal antibodies are just cells that target and destroy cancer cells in the simplest way of the immune system. Cancer vaccines are different. They're given usually, as we all know, to prevent cancer. There's been a lot of work using them and looking at them to try and treat cancer as well. Some studies showed some interesting results, but nothing has really come to pass as of yet. Finally, there are these nonspecific immunotherapies, and that's what we're all going to be talking about today. Lots of different mechanisms of action, and these nonspecific immunotherapies are, for example, the checkpoint inhibitors, which a large focus of today's talk will be on. The idea of these to release the brakes on the immune system. There are also immunomodulating drugs as well, and these are the drugs we hear lots about. This cartoon slide, in short, shows what immunotherapy is. So starting looking at one, cancer cell antigens exist on tumor cells, and they are released as cancer cells live and die through a natural process. Taking a little, what we're going to be talking about in a few minutes, it's the release of these cancer cell antigens that actually prompt the immune system. So tumors that have a lot of antigens, for example, melanoma, smoking lung cancers, and other malignancies, are the ones that tend to respond to these immunologic manipulations. After that, they have to be presented to cells called antigen-presenting cells. They then have to go to lymph nodes where the cells are primed and activated. Then they have to get to the tumors somehow, and they have to get through the blood vessel and then infiltrate into the tumors. Finally, they have to be recognized by T cells, and then they have to kill the tumor cells. And if you look on the next slide, there's probably dozens and dozens of different mechanisms of action, molecules, and targets that can help target these tumor cells. The ones we've heard about commonly and are now commercially available are the Pdl and Pd1 drugs, Pdl1 and Pdl drugs. We also have the CTLA4 drugs that are available. But if you look, there are dozens and dozens of other pathways. Some of these, for example, the IDL inhibitors, are currently in phase 2 and phase 3 clinical studies. Some of them are earlier on, and some of them are just thoughts. But a lot of these are currently being exploited or studied either in single agent or in combination now. This is a very simple cartoon looking at how these immunotherapies work. Looking at the top left slide, cancer cells secrete proteins that make lymphocytes unable to see them. Cancer cells are trying to be smart, so the immune system keeps cancer cells in check. Like antibiotics fighting bacteria, the cancer cells want to avoid them. So they put these little masks on these cartoon tumor cells, and the lymphocytes do not recognize them. And the idea is, if you look to the top right, is that these cells subsequently become activated and can attack the cancer cells. If you look on the bottom right, the mechanism of action of a lot of these drugs, these Pdl drugs, with antibodies blocking the Pdl, which is shown by black in the cartoon, are what's being bound by the drugs being given. So in essence, the cancer cells are trying to escape. They tell the lymphocytes express Pdl1. We sop up these drugs with these antibodies, and we hope that the lymphocytes attack the cancer cells. This is another cartoon looking at a very similar way. The immune system, we've simplified it before, is a very complex interaction. So on the bottom, you have a tumor cell expressing Pdl1. It interacts with a T cell, and it is made inactive by interacting Pdl1 with PD1 and others. And there are also immune cells throughout this as well. And the simple idea is if you block the Pdl1, PD1 interaction, which is what the antibodies, pembrolizumab, nivolumab, atezolizumab, and all the others of the same class do, you can then restore the T cell to help attack the tumor cell. Going back into this slide, which we've showed a couple of minutes ago, one of the things that's also being done is trying to look at multiple different pathways. CTLA-4 was actually the first FDA-approved target for immunotherapy with a drug called ipilimumab, approved in melanoma now several years ago. And it works in different fashion. On the bottom left, you can see an antigen-presenting cell activating a T cell, and the T cells are the ones trying to attack the tumors. CTLA-4, which can be upregulated in the T cell, binds this, and by doing that, the T cell is no longer activated. And on the top, and on the third part of the cartoon, you can see ipilimumab binding CTLA-4. It prohibits this interaction that was in the middle part of the slide, and lo and behold, the T cell then becomes active. Again, showing a very complicated cartoon here right now. You can see the T cell in the middle interacting with both tumor cells and dendritic cells. The take-home message here is not the molecular biology, but it's to point out that what we're trying to do now is exploit a lot of these drugs in combination. In melanoma, for example, we have combination Pdl1 and CTLA-4 drugs approved in combination, and similar studies are being done in lung cancer as well. In fact, if you look at the next slide, not only are a lot of these drugs being used as single agents, a lot of these drugs are now being used in combination in studies. For example, IDO inhibitors on the bottom right are being combined with PDL drugs and other drugs as well, trying to harness multiple different aspects of the immune system. Two things I just want to mention here. As you look on the slide, one thing that you can see is that as these immune cells bulk up, they attack the tumor. And one of these phenomena that you can see is what's called pseudo progression. Pseudo progression is just that. The cancer looks like it's getting worse on a scan. And that's because the first thing that happens before these tumor cells die is you get an infiltration of immune cells. So if you look at a scan, it can actually look worse on a scan. It's a very important concept for both physicians and patients to understand, because one of the things we often look at is size of the tumor on imaging. So one has to look at that very closely. One thing also to mention is that as we think about the side effects of all these different drugs, we get concerned about autoimmune phenomenon or attacking the body's own cells. The body lives in a very fine balance of what these immune cells are trying to do. By turning off the immune system, one thing that you'll often see in this pathway is not only does it attack the tumor, but it attacks self. And as you'll hear later on, as we talk about side effects, the side effects seen with these drugs in general are autoimmune in nature. Finally, a talk in lung cancer wouldn't be complete without talking about the newest things that are available, what's called CAR T therapy, or chimeric antigen T cells. While it's not yet approved or really close to being approved in lung cancer, I think not a day goes by that in clinic we're not all asked about these questions, is where can I get CAR T for my lung cancer? And it's one of the most fascinating things known. What happens is, as we've talked about before, the patient's immune system doesn't recognize antigens on a tumor cell. So what is done is T cells are actually harvested out of the patient's body, which is a completely novel concept. Those T cells are then transfected with a viral vector. And the viral vector does a couple of things, but most importantly, it has a molecule on there that recognizes the antigens on tumor cells. As you can see on this slide, those cells are then transfected in a lab on the top right, and then they're injected back in. And two things happen. One is the T cells propagate, they grow, and they attack the malignant cells as well. This is completely different from what oncologists are used to doing. And although this is a little bit of a far-fetched dream from approval today for lung cancer, there are many studies in solid tumors, including lung cancer studies throughout the world. The patient's T cells are harvested, as you can see on the top left. They're actually engineered in a cellular engineering lab, which obviously is expensive and complicated. That cell product is harvested and they're reinfused back. And these T cells now have viral vectors in them, so they A, propagate, and B, recognize the tumor cells. This is a slide from the FDA approval of the first CAR T therapy for leukemia. And not wanting to go into super details, but the most fascinating thing is on the bottom right. On the bottom of the slide. And what happens is these cells are injected into the patient, and not only are they there, but they grow. So if you look at the bottom on day 0, and they, over time, between 0 and 1 month, you get over 100,000 times the number of cells originally started with. And eventually, over time, it peters out. However, as you can see, over the long term, these cells still exist. So first, you get a rapid attacking of the tumor. That leads to the consequences of these cells because they activate the immune system, and the biggest side effect is cytokine release syndrome. But the true exciting aspect is that these cells, after a single or 2 infusions, linger for months and potentially years. And the idea is they can keep the cancer in check. The side effects are real. Some new modalities being done to mitigate some of the major side effects are to temporarily transfect them with not permanent viral vectors, but smaller molecules, so the effects are not as profound and they don't last as long. And then secondly, to not fully activate the immune system with T cells, but to activate them with other molecules. I call that CART light. And what's happening is there's an antibody that attacks the cells, but it's bringing in the T cells directly as well. So in short, there are multiple different places in the immune system for targeting, all of which are very, very different from each other, but they're also being used in combination. We currently have 2 approved mechanisms of action, PDL and CTLA-4. Most of the time, we're using them as single agent, but we expect a lot of combinations to be approved soon. And then finally, there are multiple novel mechanisms also. Thank you.
Thank you, Alex, for bringing forward a pretty complicated mechanism, but helping us reach some understanding for how immunotherapy works. I'll now ask us to submit any questions, either online in the box below, or again, a reminder to our in-person audience to please use index cards at the table. I think we have some questions.
We do. And Dr. Levy might be addressing this, so I'm just going to ask them, and you can let us know. We have a question: what emerging biomarker assays are available or becoming available for selection of personalized IO combinations?
So I think Ben will talk about that, but Alex, if you want to jump in a little.
Yeah, and I should have mentioned a little bit more in my talk. So the most common thing that we look at now is Pdl One Staining. As you can see, if you on those slides, if you have a higher Pdl One staining, it's more likely that the immune system is involved in the tumors. The problem with that is it's not a very good test. We currently use numbers from 0 to 100%, and we think 50 is better. Greater than 50 is better than 0, but it's not the best test that we have. And as we all know, or patients and physicians read, there's multiple different assays. One of the most important tools that we have going forward is what's called tumor mutational burden. From my initial slide, you saw the way this all works is by having tumor antigens being expressed on tumor cells. So the more antigens you have, which are usually mutant proteins or overexpressed proteins, the more likely these cells are to be immunogenic. So the use of this new assay called tumor mutational burden, not quite ready for prime time. It takes, you can order it, but it takes a lot of time, lengthy time to come back, and we still don't fully understand it, but that's probably going to be the most important thing that we have going forward.
And then we have a few questions about how well immunotherapy works for EGFR positive patients.
So I think Ben will also touch on this, but maybe briefly, Alex, your thoughts about using immunotherapy for an EGFR mutant patient.
So this is something that I think if you ask 100 thoracic medical oncologists, you'll probably get 200 opinions, probably. So in general, it doesn't work very well. And if you look at some of the best data that we have, really meta-analysis and looking at all the different studies, the response rates are probably not better than chemotherapy. And there's 2 real views for that. One is, should you just give it instead of chemo, or should you just give chemo? The real fundamental issue is that if you look very closely at it, this whole process works by tumor antigens and overexpression of mutant proteins. And the EGFR mutant population usually has a single driver of causing that cancer to occur. So with that in mind, these tumors are not very immunogenic. We've all had patients where you have a EGFR mutation but a high PDL status. And you wonder what to do in that situation because one biomarker tells you to do one thing, but you know it's probably not going to work very well. I think most of us who see a lot of these patients will tell you, based upon our own clinical experience as well as a wealth of the data, is that single agent immunotherapy does not work very well in the EGFR or any driver mutation driven cancer, ALK, BRAF, et cetera. They're all the same. I think the real test will be, how do we make these cancers more immunogenic? How do we drive that immune response to the cancer? Can you find that single antigen? Can you drive it to the immune cells? And it's going to be tough, but I think that was a long answer to say it doesn't really work very well.
Yeah, I think that tying those 2 questions together, because we don't have a great positive predictive marker, it's really difficult to select who those patients are. the likelihood of success is very low. Not to say that it's not possible, not to say that it's not an option, but in many cases there may be better options. I think Dr. Levy will go over that in a little more detail.